I'm Dr. Kathuria, and welcome to our podcast, Conversations with an Organ Engineer. Today, we'll talk about personalized medicine and how it applies to tissue engineering and organoid development.
Hello everybody, welcome to Conversations with an Organ Engineer, where our organ engineer is Dr. Kathuria. My name is Alberto Bustamante. I'm an incoming second year master's student here at Johns Hopkins University, and we have a different member today. You want to introduce yourself?
Hi everyone, I'm Kai. I'm a second year master's student in biomedical engineering here at Johns Hopkins. So I'm here working in Dr. Kathuria's lab, mostly focused on computational side, dealing with MEA data.
Hey, yes.
Hi everyone, I'm back again. I guess you know us from the last video, so I will not introduce myself all over again. So go back and watch our previous video. And today we're going to be talking about personalized medicine that we touched upon last time. So
for me, when I think of personalized medicine, I think of, everyone's body. We all have the same human anatomy, right?
Yep. And
we all on a basis we work the same way, but we can also be very specific and specialized. Not everyone has the same blood type. Not everyone has the same genes. So with this concept of personalized medicine, I think it's taking what we know and the workings of the human body and tuning it to be on an individual basis, right?
Correct. So personalized medicine comes from the concept of everybody's anatomy is the same, the human body, as you put it, is the same, but we have had different upbringings. Different socioeconomic backgrounds, different cultures, So, for example, a kid who has grown up in India will be on a rice and a wheat diet. This is an example, but a person or a kid who has grown up in the American diet system will have pizza, pasta, bread,
American food
and hamburgers as their diet. And the diet itself causes epigenetic and genetics changes because that's when you're developing and your body develops differently. So the idea would be, because everybody has these subtle growth differences during development, the idea was that your body will react differently to a different drug. And this is very specific to mental health, particularly. With diseases such as schizophrenia, bipolar, depression, anxiety, and God knows who doesn't have anxiety. Everybody has anxiety. I've heard from a 5-year-old that I have anxiety.
You're 5.
And I'm like, you're 5 years old. But they have stresses. Everybody has stresses, and that leads to anxiety. And anxiety manifests, and then it goes into these serious mental health disorders. And not one size fits all doesn't happen in the psychiatric world. So that's what we're going to talk about in this discussion today and today's podcast.
Now, I want to circle back to it because you said something interesting to me. You brought up epigenetics.
Yeah.
And for me, when I think of, or from what I can grasp, there's other factors other than our genetics and stuff like that impact our cells and our genes and how they behave. Would you say that epigenetics can play a lot in tissue engineering? I mean, I know on a cellular basis it does, but how do you scale that up to tissue engineering?
So first we'd have to define the concept of epigenetics. For those who don't know what epigenetics is, epigenetics So you have all your genes. There's about, I don't know the exact number off the top of my head, but there's several types of genes that we have in our body. They're expressed. But think of it, not every gene is required to be expressed at every time. And so body likes to conserve energy. So we have something called chromatin. Chromatin is like the structure around the genes that wraps around the genes. And you have little like switchboards. Epigenetics is like switches on your genes. That's the best example I can give of a very relatable example. There's an environmental trigger that switches on, and then that's your epigenetic signature. So it will say, now turn this gene on because we need this protein, because we need this function for the body to perform. So that's how it goes in chains. And so epigenetics has a huge role in personalized medicine. And why does that have the role? As I already said, your environment plays a huge role on how the body develops physiologically. And those physiological reactions have a tendency in your drug selection when you are going through any disease of any kind, any treatment, diagnosis, and prevention. We can decide on that basis. But in terms of organ engineering and tissue engineering, we talked about stem cells and iPSCs in our last podcast. With induced pluripotent stem cells, when we reprogram them or get back to their naive state, they lose their epigenetic signature. So it gets a blank slate.
Clean slate, yeah.
Clean slate, which is a sad part of this programming. So, but the genetic signature is still maintained. So I still carry a genetic profile, but I lose my epigenetic signature, which is the sad part. So I can't carry all the environmental factors. But I can understand with your genetic profile, how would you react to glucose? So if I give you high doses of glucose and your family has a tendency of insulin resistance, then my cells would react the same way.
So when you say stem cells and they lose their epigenetic factors, they lose it to some extent. Like they say if we, like skin cells, right? They would lose the specific characteristics of skin, but not of like who we are in general.
No, they won't lose your DNA profile. You carry those 23 pairs of chromosomes. We have to check as one of the things of quality checks when we make these stem cells and reprogram them from the skin cell to the stem cell. We do check for all the 23 chromosomes. So you carry that no matter what. And so you do carry your personal signature of the genes, but not the epigenetics, because we kind of wipe that slate clean.
Yeah. And I just also, something that I think is pretty interesting, and this kind of crosses a little bit into like psychology, is this with this like nature and nurture type of thing. I feel like you can, epigenetics and cell development can relate to that in some extent. Yeah.
Nurture side of it. And the nature side of it is the genetics, I would say. That would be my opinion. There's varying opinions in the scientific world on this, but yes, that would be my opinion.
Okay.
I was going to ask, when you when you actually do like reprogramming, how do you specifically how do you wipe out those environmental factors?
It's just a byproduct of the reprogramming. It's a byproduct of the reprogramming.
Oh, like it happens naturally.
It happens naturally. It's a byproduct. When we reprogram the skin cells to stem cells, it's a byproduct of reprogramming the process itself, that it wipes the environmental or epigenetic markers.
Okay. So,
because why is that we're going back into time for that cell. We're changing it from what it was differentiated as a fibroblast, a skin cell, to an iPS state, which is like a baby. right? So baby is not exposed to a lot of things. So it's wiping out all the environmental signatures that it has seen over time.
Okay. Because I was thinking about since they are, like when you derive them from, let's say from the patient, they already like start to have like some protein like expression for a long time.
Yeah.
So when you reprogramming them, it's just like gone away. Like
yeah, it's wiping the slate clean naturally happens because of this state, because it goes from a differentiated cell to a non-differentiated cell, which is a stem cell is always like, I always compare it to like a baby or a naive state where it doesn't know what it wants to be, and so it wipes all the things that it's seen in the past, and philosophically it turns into where it did some time travel. Yeah.
Okay.
And or wipe the slate clean, as Alberta pointed out. But one of the things that's interesting, and now it's not science, just philosophy, I wish we could do it in life. Wipe slate cleans. Okay.
Now, when you wipe these epigenetic factors, is it based on a certain time that you need to let like your sample stay or like be exposed to certain like transcriptional factors to wipe them, or is it like once they're exposed, it doesn't matter how like the time that it's been exposed, as long as they're interacted or they're exposed to those factors?
So what we do is use the 4 transcription factors that were defined by Yamanaka and his group in 2007, 2008. And then they subsequently won the Nobel Prize in 2012 for it, late 2012. I can name the transcription factors.
It's like
SOX2, CMYC, KIF4, KLF4, and there's one more. And those 4, basically, when you put them back into the cell, those transcription factors, they express the genes that are required to revert the age. And yes, so that as soon as that transcription factors are put in, they wipe as soon as they come in contact.
Okay.
So you are right. As soon as they come in contact, when they start expressing those genes, they revert back to a stem cell state.
And I remember from the first episode, you said that with differentiating stem cells, they kind of need guidance, right? They need a push to put them in the right direction if we want them to become a brain cell or a lung cell or any type of cell. And to guide them, I assume we're utilizing these transcriptional factors to guide them there? Or?
No, for guiding them into a different cell type, we use chemicals.
Okay.
We do chemical differentiation in our lab. There's various ways of doing this. We use chemicals to direct them. So we add different proteins, different chemical structures at different times to direct the differentiation. We're basically trying to manipulate the body when it's in the embryonic state. So, for example, to turn brain cells, we have to stop this pathway that is called SMAD inhibition, which this pathway, when it stops, it directs the stem cells into the ectoderm layer, which is the precursor of the neural tube formation. So this is how we do it in-house when we're making brain cells. So for those who don't understand just what I said, it's the embryonic layers there's 3 layers, 3 germ layers. So ectoderm, endoderm, mesoderm. And each layer forms different organs. And for us, it's the ectoderm that forms the brain.
Okay. To bring it back to personalization, one thing that came to my mind is bone marrow transplantation, where this treatment is used for, I think, sickle cell anemia, for leukemia and other types of blood cancers or illnesses or diseases like that, where essentially you're gathering the cells of your patient, right? You're manipulating them in such a way where you encourage healthy stem cells, and you're infusing them back to the patient to improve the cells that they have that might be detrimental to their system. Does personalized medicine always look like that process, or I guess how different can it be and still be personalized?
Personalized medicine can be very different for different people. For cancer, it's different. I was talking about mental health, particularly, and neuropsychiatric disorders is very different. And for, let's say, diabetes and endocrinological disorders, it is different. So depending on the disease, it's different. It's a little difficult for it's more useful, I would say, not difficult, it's more useful for neurological diseases because we have very little knowledge of the brain, and particularly the diagnosis of mental health is very variable. We don't have a particular biomarker for it, and so that's why personalized medicine becomes more legitimate in the cases of mental health and neurological disorders. But for endocrinology and endocrinological disorders like type 1 diabetes, type 2 diabetes, we know the insulin doesn't work properly. We have different disease drugs that people have been prescribed for ages and they have been doing well, like for example metformin for type 1 diabetes and type 2 diabetes. We get the insulin pumps and all of that has been working. But now with people focusing on diversity, inclusion, and equity these days, we have also pushed forward towards the concept of not just looking at one person or one type of person, but looking at multiple people. Like for diabetes, African American people have a high incidence and they react differently to drugs than Asian people would, or a Caucasian person would. So this is where personalized medicine comes in very, very useful, because we can, using your individuality, your environmental upbringing, your socioeconomic status, those developmental factors for your body and physiological conditions are different than someone else.
Right. So there's layers to it.
Yes.
Right. So at the base, we're all human, right?
Yeah.
We all have the same anatomy to some extent. And then after, there's also specific regions of, or ethnic groups, right?
Yep, ethnic groups,
age,
socioeconomic backgrounds comes into play. As you said, age, gender, socioeconomic, ethnic background, and then your diet comes into play, which is like the uppermost layer, which people don't even think about.
Yeah. No, that makes sense.
Fitness level comes into play, and that can be personalized too, because someone who runs a marathon every 6 months to a person who's very sedentary will have a different response to a different medication. And you will notice health and fitness plays a very huge role again in personalized medicine too, which is very surprising to people. People get that shock. What? No. I was like, but your epigenetic factors are regulated by how much you exercise or what you eat, how many cookies you had the day before. And not that I'm saying not to have cookies. I'm all for cookies. Yes. But we also have them in the lab, just FYI.
I love the pecan.
See, he already said he loves the pecan. So we're not saying that is against having cookies. It's just
it impacts.
It influences the decision of the doctor to say, okay, I'm going to put you on this dose rather on that dose. And as I said, your socioeconomic background also helps.
Yeah.
Because let's say you are from an affluent family and they're very regular about diet, exercise, and everything, so you eat healthier. But if your socioeconomic background is a poor one, your eating habits, your diet, your sleep patterns will all change.
Yeah.
And so my medication level for you as a doctor would be different.
Yeah.
So it all depends on all the factors
Your
that we
groups and all
listed.
that.
And that's why personalized medicine is now being encouraged, and I would suggest is to ask about it from your physician. What can you do? What is personalized to me? And with organ engineering, what we're trying to do, where we come in, we can take your stem cells, convert into the organ that we think you're having a problem with. So, for example, if we're talking about mental health and mental health disorders, where I see personalized medicine gaining momentum is, let's say Kai we're not saying he has a particular diagnosis, but hypothetically, I suffer for anxiety. For example, I suffer for anxiety, and I want to look at what does my brain do wrong compared to Kai's, who's very healthy and does not suffer from anxiety. And so I derive my skin cells into brain organoids. I start looking at all the drugs that are in the market, and from clozapine to Prozac to whatever there exist. And I find that this particular dose at this particular time period, if given, and this disease or one or more of these drugs helps the brain react better. And so I can provide that information to a clinician, and he can tell you as a clinical decision support report that will be useful for the clinician to see and say, okay, this person reacts to this medication and this dose, so I better give him this one. Or he reacts to these 5 doses, and I think with his clinical judgment, okay, out of those 5 diseases, 5 drugs that I saw, this might be the best dose, so I might give him that one.
Okay.
And that would be the clinician's discretion. But at least with that information, he is more empowered to make the right medication call with the right dose call, without for the patient suffering for 6 to 8 months trying hit and trial. Oh, give this drug today.
Trial and error. Yeah, it can be scary.
Or that drug, and I'm in a depressive mood for the next 6 months because the drug did not suit me, and I switched physicians. And I keep switching physicians until the end of time because this drug isn't working for me.
Okay. And I think that's very important what you said, and I can relate to that because a while ago I was placed on a drug, right? It was a specific oral steroid, and for an allergic reaction that I was having. But it impacted me in such a severe way. I just went back to the doctor. I was like, I can't be on this anymore. Is there an alternative? And they put me on a different one, and it was like night and day. Like it had such a different effect on me. It was like smoother. So it's just, to me, it's amazing how our personalization of our biology goes so far, and different drugs and different treatments, they really do impact us differently.
Yeah, and again, this brings us to the topic of how we can spread this knowledge further and further, and that's what our aim is with this podcast, is to engage with the audience and get some feedback. So for our next episode, we would like for you to give us comments on what do you think about personalized medicine, how we can help. Please email us.
Our email is convosworganengineer@gmail.com. So CONVOSWORGAN ENGINEER.
And he will put it in the subtitle somewhere in the comment section or title section, because otherwise I cannot memorize it.
And we just want to say thank you to the subscribers that I think we have 17. Thank you so much. And the 4 people that left comments on our first episode. You guys are very nice. We appreciate the kind words.
DM us for questions, please.
Yeah.